Hello, I am Sarah Jane Demy, and on behalf of the Demy-Colton team and our sponsor for this virtual salon, WBB Securities and Research, it is my pleasure to welcome you to the 42nd installment of the Demy-Colton Virtual Salon Series. As always, we welcome past participants back, and we welcome all of you who are new, who are joining us for the very first time. As many of you know, Demy-Colton's mission is to convene healthcare industry leaders to catalyze change. And we do this through our virtual and in-person events, including BioFuture, taking place here in a very busy New York City, October 4 to 6. Biotech Showcase, which will be held next January in San Francisco, and our CEO Summits, invitation-only events, which just took place in Barcelona and La Jolla. And last but not least, our virtual salon series. The topic of today's salon is exploring solid tumor treatment and its cure, a conversation with prominent KOLs and its future. Before we get underway, I do want to thank our sponsor, once again, WBB Securities and Research, for sponsoring this truly important conversation. Before I turn the virtual mic over, please note that the salon will be a bit different than our usual. We will begin with a short introduction video on the advancement and treatment of solid tumors, followed by a general discussion among the panelists, which will last around 45 minutes, and then we will move on to audience Q&A. Please note that this salon is being recorded and it will be available for on-demand viewing later today. And now, I am pleased to introduce our sponsor and one of our moderators, Steve Brozak. Steve is Managing Partner and President of WBB Securities, who will introduce our opening video. Steve.
Thank you, Sarah, and the Demy-Colton team for organizing this important salon. Good morning, and for those overseas viewers, good afternoon or good evening. I'm Steve Brozak, WBB Research Institute's founder. Since our inception, our mission has been to examine critical real-world issues at the intersection of healthcare and the life sciences industry. In keeping with our mission, we are very pleased to introduce this presentation examining IRGD peptides and their real-world oncology use in treating solid tumors. It now gives me great pleasure to have last year's Lasker Prize winner, Dr. Erkki Ruoslahti, who will be explaining his discovery work with integrins and RGD. To help us with an overview, we begin with a video synopsis of the current status and the recent advances in cancer therapy. This is narrated by the Lisata Chief Medical Officer, Dr. Kristen Buck. The video, please.
Hi, I'm Dr. Kristen Buck, the Chief Medical Officer and Head of Research and Development at Lisata Therapeutics. It is my pleasure to describe a new advancement in the treatment of solid tumors. Most people recognize that despite recent advances in care, cancer is still the #1 cause of death worldwide. As such, it represents a significant unmet medical need. Interestingly, and perhaps not well recognized, solid tumors account for over 90% of newly diagnosed cancer. And while there has been notable advancement in the treatment and cure of leukemia and other non-solid tumors, such as multiple myeloma, similar successes in the treatment of solid tumors, for example, pancreatic, gastric, and glioblastoma multiforme, brain cancer, have not kept pace. Solid tumors are often inadequately treated with our current treatment paradigms for two main reasons. #1, ineffective tumor targeting, and #2, ineffective tumoral penetration. This occurs because most solid tumors have a physical barrier surrounding them called the stroma, which acts to inhibit the penetration of anti-cancer drugs. Additionally, solid tumors have a hostile tumor microenvironment, which includes immunosuppressive T cells. The tumor microenvironment contributes to treatment resistance and promotes metastases, or the spread of the tumor throughout the body. And while we may have effective targeted therapies which get anti-cancer drugs to the tumor, the stroma still often impedes penetration into the tumor. Furthermore, non-selective penetration enhancers are, by definition, indiscriminate and increase the likelihood of anti-cancer agent penetrating non-cancerous tissue, causing what is known as off-target side effects. Thus, escalating doses of anti-cancer drugs often results in drug discontinuation due to such intolerable side effects. The last challenge in treating and curing solid tumors is that while animal models aid us in understanding the biology of cancer and aid us for screening new agents, human cancer biology is often more complicated than animals, thus not fully translatable. Recent research has led to the discovery of a naturally occurring mechanism which may help optimize drug delivery into solid tumors called the SEND-R active transport mechanism. This mechanism helps ferry anti-cancer drugs across cells and into the body of the tumor. RGD peptides have been discovered and contain the amino acid sequence arginine, glycine, and aspartic acid. RGD peptides alone are tumor targeting agents, that is, they home to solid tumors, but they don't enhance tumor penetration. RGD peptides or internalizing RGD peptides, however, have the special sauce. These were discovered by Dr. Erkki Ruoslahti and colleagues and have the ability to specifically target solid tumors and enhance penetration, the latter being activated by the SEND-R transport mechanism. LISTA-1, a novel IRGD cyclic peptide, works through the SEND-R platform to specifically target tumors and convert their stroma from a barrier to a conduit. Interestingly, due to its mechanism of action, it can be co-administered with most anti-cancer standard of care drugs or tethered to such drugs by molecular bonding. Furthermore, LISTA-1 works with and is completely agnostic to the modality or modalities of standard of care agents, be those cytotoxics, immunotherapies, or RNA agents. LISTA-1 also selectively depletes intratumoral immunosuppressive cells, thereby combating anti-cancer agent resistance. In work performed by Dr. Sugahara and colleagues, LISTA-1 has been shown preclinically in highly fibrotic tumors to inhibit the metastatic cascade. Now I will describe the novel mechanism of action of LISTA-1, an internalizing RGD peptide. As you can see, it's a 9-amino acid cyclic peptide with high affinity and selective binding to alpha V beta 3 and alpha V beta 5 integrins, which are upregulated on tumor vascular endothelial cells and tumor cells. Once bound to these integrins, LISTA-1 is cleaved by naturally occurring proteases or protein enzymes such as furin to form a C-end rule linear peptide or SEND-R peptide. The SEND-R peptide then goes on to have high binding affinity for neuropilin-1, an adjacent receptor also upregulated in solid tumors. Upon neuropilin-1 activation, the SEND-R transport pathway is initiated, forming microvesicles at the surface of the tumor. The microvesicles engulf anti-cancer drugs in the circulation and pull them through the stroma and down into the tumor. This mechanism has been validated in clinical studies. The results of phase 1 studies with LISTA-1 suggest that the effects of this internalizing RGD peptide translate well from animal models to humans. This further gives hope that the exploitation of the SEND-R transport mechanism, a tumor targeting and penetration approach, will bring important advancement to the treatment of advanced solid tumors.
Thank you for that. So now it is my pleasure to introduce the other moderator for today's virtual salon, Annette Brendel. She is the managing editor of BioWorld Science and is joining us today from Hamburg, Germany. Annette will be joined by our renowned panelist, Erkki Ruoslahti, who among all of his achievements was recently awarded the Albert Lasker Basic Medical Research Award. And Erkki is joining us from California. Kazuki Sugahara. Kazuki is an assistant professor of surgery at Columbia University here in New York. Andrew Dean, who is the head of the Department of Medical Oncology at the St. John of God Sebago Hospital in Perth, Australia. So it's very late for Andrew. And David Matzo, he is CEO of Lasada Therapeutics, located in New Jersey. So he's close to us. So without further ado, it is my pleasure to turn the virtual podium over to Annette. And Steve, you'll stay on with Annette, right?
Yes.
Okay, wonderful.
All right, wonderful. Hello, everybody. Good morning and afternoon and evening. Thank you to all the panelists for joining us today, and to Demy-Colton for putting on this important discussion. So I want to start with a few questions for Dr. Ruoslahti. Dr. Ruoslahti, the first time we talked, I was a cub reporter at BioWorld, and you had just won the Kyoto Prize. By then, since then, you have won, a lot of accolades. So congratulations on the Lasker Award last year, which is the most recent one to my knowledge. Can you tell our audience how you got interested in RGDs and a little bit about your scientific journey there?
Yes. So. The question I started studying was to see how normal cells know where they should be and how cancer cells do not obey the rules and metastasize in various places. And nothing was known about what might direct these events at the molecular level. So we then ended up finding some of the first integrins and the RGD sequence, which many integrins bind to in their target proteins, particularly in the extracellular matrix. And at the same time, two other laboratories coming from a different angle to this same problem found their favorite integrins, and we that's how we ended up sharing the Lasker Prize. Now the IRGD is a different story than the RGD itself, because the I that we put there is for internalizing. This is a special RGD peptide that gets into the cells, and as we'll see also already in the introduction, it also
All right. Unfortunately, we seem to have lost Dr. Ruoslahti. So perhaps we should just, hopefully he will be back in a little bit. But I was going to be asking about the internalizing RGD. So perhaps Dr. Sugahara, you can answer some questions about that. Can you tell us a little bit about how the IRGD is different from regular RGDs, and how that might be important for its clinical development? Because I think the potential of RGDs was quickly obvious when Dr. Ruoslahti and the other labs he mentioned discovered them, but early attempts were not successful in the clinic. So can you take us through that story a little bit?
Sure. So the original RGDs, they bind to proteins to inhibit the interaction with integrins. And some integrins are important for cell survival. So by inhibiting the interaction, some of us thought that we can kill some cells. Another way is that we can use the RGD peptide to deliver things to tumor blood vessels, because there are certain integrins that interact with the RGD peptide. However, the downside was that the RGD peptide would get to the tumor, but it does not spread, and the effect was very minimal. On the other hand, the IRGD peptide does a little bit more than usual RGD peptides. because it binds to neuropilin, the second receptor, in addition to the integrins, it can now spread all over into the tumor to deliver things really deeply, widely throughout the tumor. That's the biggest difference between the RGD peptide.
And is neuropilin expressed on the tumor cells itself?
Yes, so neuropilin, interestingly, is expressed in all sorts of cells, but is upregulated especially in tumor cells and the blood vessels in the tumors.
Mm-hmm.
And the combination of the integrins and the proteases that cleave the peptide so that it can bind to neuropilin and the high expression of the neuropilin, that combination makes the peptide very tumor specific.
Okay. And so in the introductory video, I perked up when I heard that the Listot, the specific IRGD that we'll be talking about a little bit more now and later in the program, can be either co-administered or tethered to other drugs. Like, and when I hear tethered to other drugs, I think antibody drug conjugates. Can you tell our readers, maybe especially our not our readers, apologies, you can tell I write more than I talk. So can you tell our listeners, especially perhaps the ones who are not intimately familiar with LISTA-1 and this RGD approach, how LISTA-1 is similar to and different from antibody drug conjugates?
Yeah, let me start from the mechanism standpoint, the penetration mechanism. Just like we saw in the video, the penetration depends on a pathway that the peptide creates within the tumor tissue. And it was very important for us to understand that mechanism because just like we have been talking, we knew from all the phenomena that we saw in animals that the peptide would somehow target tumors very specifically. It would even spread within the tumor, and it would even deliver drugs that are not even conjugated or connected to the peptide. So it was almost like a magic. So when we first discovered it, some people said, oh, that's great, but really? So as we understood the mechanism, we now know that there is a reason that it works. It's not a magic. And the pathway that was described is exactly it, that it creates a tunnel through cells and into the tissue so that not only the peptide and drugs connected to the peptide can go into the tumor, but also things that are around the tumor, around the peptide, can also go in. And that makes a big difference between antibody drug conjugates, because in that system, drugs are conjugated to the antibody, which usually cannot spread into tumors or cannot deliver things that are co-injected with them. The peptide that we discovered can do both. It spreads and it can deliver things without conjugation. So that's a big difference.
Okay. So tell me specifically how LISTA-1 is able to interfere with the metastatic cascade, or what you think is the mechanism there, because metastasis, of course, like to my knowledge, is still what kills the majority of cancer patients, right? And it is a very general issue, also very broadly important across solid tumors. And I'd love to know, how you think LISTA-1 could have success there, based on, your experiments, your own research.
Yeah. Right. To be honest, we don't know the mechanism 100% yet, but we know some of it. Some time ago, we first demonstrated that the peptide alone can inhibit metastasis, for example, in pancreas cancer mice, breast cancer mice, prostate cancer mice. At that point, our understanding was that the peptide interferes with neuropilin-1, the second receptor that we have been talking about. And that causes a stall in the motility of tumor cells so that the tumor cells can no longer spread or move. That was the initial mechanism that we found. But in our most recent study, which actually can be already read in a bioarchive, we're still trying to publish it. There, we found an even stronger effect that interferes with the metastasis. In some tumor models, we found that the peptide alone completely inhibited metastasis to the liver or to the lung. There, we think that there is some more mechanism to what we had discovered before, which maybe involves the immune system. As briefly mentioned in the video, we now know that the peptide works as an immunomodulator that turns a cold tumor into a hot one to have a more activated anti-tumor immunity. And that might have a role in the anti-metastatic effects, but we're working on it now.
Okay, so that is fascinating. And tell me a little bit more about this immunomodulatory role, because tumor immunotherapy is, you know, I would say the most recent major breakthrough, and is now, you know, has very rapidly become like one pillar of cancer treatment. So, but also, as most of us know, it doesn't always work. And one of the big goals, I would say, of current anti-tumor therapies are to increase the efficacy and to, you know, get more patients to be able to benefit there. So how do you think this is working as an immunomodulator? And is it affecting the immune cells? Is it affecting the tumor cells? Is it affecting the stroma? What do you think?
Yes. So you're exactly right that many tumors do not respond well to immunotherapy yet. And what the IRGD peptide does is that it reduces the cells, the immune cells, that suppress anti-tumor immunity. So, for example, in pancreatic cancer, there are those negative immune cells that protect the tumor from our immune system so that the tumor can grow. And we had to overcome that mechanism in some way. What we found was that our peptide significantly decreases those negative immune cells so that the active ones can start targeting the tumor cells. And that's what the peptide does. And when we combine with some of the immunotherapy agents, now we can tell that those agents work better when we combine with the IRGD peptide.
Okay, so that is fascinating. Are you combining it specifically with checkpoint blockers or with you know, cellular therapies, which are also being tested now? You know, CAR T cells, of course, still mainly you know, mainly effective in blood cancers, but slowly making their way into the solid tumor space. What do you think? You know, what are you testing, and do you have like thoughts or hopes on what the eventual landscape might look like?
Yeah. So in our hands, we have been combining the peptide or peptide-based chemotherapy with immune checkpoint inhibitors. So we have a good feel that it's probably going to work in humans too, and maybe Dr. Dean Andrew can tell us a little bit about it later. But there are also reports where people have tried to deliver T cells using the peptide to enhance anti-tumor immunity. and some people have succeeded in it. So I think that can be an option for the future.
Okay. All right, and so I would love to hear Dr. Dean actually weigh in on this, because LISTA-1 is also in clinical development, and Dr. Dean is leading those efforts. So can you tell us a little bit about, as a clinical oncologist treating solid tumors, what do you see as your biggest current problem to treating and curing cancers? How do you think our current drugs can be made more effective for more people?
Well, I think that, thanks very much, Annette. It's a very good question, but I guess in an ideal world, you want to create a perfect storm of something that damages the cancer cells, that gets the cancer cells to meet the immune system, and then obviously get some drugs that actually get the immune system engaged against the cancer. And I think the LISTA-1 compound is showing some great promise in some of the very early phase trials that we're doing. So the original trial that we did with LISTA-1, which was called the SEND trial, that showed the best ever response rate seen in a pancreatic cancer trial, where 26 of the 29 patients who took part in that study actually showed a reduction in size of the tumor, and that's not something that you normally see with pancreatic cancer. It's something that you see with things like breast cancer, Yeah. And so I think when we were first approached to trial the compound, the science was sounded fantastic, and the question was, would the molecule actually live up to the promise? And I can say it absolutely did. And so for us, the next step was to say, well, if the original science is correct, that LISTA-1 opens up the vesicles in the blood vessel wall, which actively transport chemotherapy across into the tumor, then we're overcoming one of the major barriers, which is this sort of protective layer that cancer cells can grow around themselves, called the tumor microenvironment, or stroma. And this acts as a protection against the chemotherapy drugs. So if LISTA-1 overcomes that, what else can we use it to get into the tumor? And so the basic science suggests that LISTA-1 loosens intercellular connections, which allows tumor infiltrating lymphocytes to pass from the blood vessels into the tumor. So we've developed a new study called iLISTA, short for immune LISTA, and we're combining standard chemotherapy with gemcitabine and nab-paclitaxel. We're using LISTA-1 to enhance the delivery of the drugs into the tumor, and we're also adding in an immune therapy molecule, durvalumab, as the data suggests that the immune therapy molecule will likewise be transported into the tumor microenvironment. And what I can share with you is just some very early observations that all 3 of the first patients on that particular study, who have had a second biopsy taken at the 12-week mark, so 12 weeks after starting the combination therapy, all 3 out of 3 have got tumor infiltrating lymphocytes present in pancreatic cancer. And in fact, one of the participants has had an almost complete disappearance of the pancreatic tumor, which has just been replaced by fibrosis, scar tissue, and just a mass of immune cells. So, as a clinician, I sincerely hope that we're actually on the verge of creating this perfect storm of chemotherapy, immunotherapy, all being enhanced by LISTA-1 to savage the tumors wherever they may be.
That is, those are impressive response rates, I have to say. That is not, something you normally hear with pancreatic cancer. Can you talk a little bit, about safety issues and how specific this is to the tumor? Because, integrins are a very, broadly, they're on all cell types throughout the body, of course. And this idea of opening up blood vessels is great if you do it in the tumor. But, if you're doing it non-specifically, you're going to have another problem. So can you talk about, and both, maybe both your clinical experience and Dr. Sugahara, if you have any research there that speaks to why this would be specific? So I'm curious what you saw and what you would expect to see there in terms of specificity and safety of the approach.
So I guess from my perspective,
maybe I can start with.
I was going to say I get the easy bit, which is that the scientists do all the hard work, and once they've found the solution to all our problems, we just get to try it out and show that it works. So. From my point of view, the fact that it transports antibodies that are the right molecular weight into the mix, the fact that it transports chemotherapy drugs, I think as an oncologist, the potential for this compound is sensational. There's great data showing that it's extremely safe for normal tissues, because normal tissues do not overexpress the required receptors for the protein, for the peptide. And the cells that we're really interested in, as in the cancer cells, dramatically overexpress these the receptors. So the early phase data, which I'm sure Kazuki will be able to explain in far better terms than me, that you can get vastly enhanced levels of chemotherapy, 13, 14, 15 times concentration of chemotherapy drug pushed into the tumor cells with the compound. And with the antibodies, for instance, with trastuzumab, I believe the figure was 40 times the tumor-killing antibody trastuzumab was pushed into the cells compared to normal. So, Kazuki, what do you think?
Yes, I think that was an excellent answer. And for example, for trastuzumab, like those antibodies, they have really good binding ability to their target. It's too good that once they meet their target, they cannot make their way through. They're just stuck. So antibodies in general, they have very nice specificity, but they cannot spread. And that's what the peptide can help. And imagine you being the IRGD peptide being injected into the bloodstream. So now you're in a tunnel of the blood vessel, only seeing inside the blood vessel. So if you're in that environment, the only area where the integrins are expressed is the tumor blood vessel. In normal blood vessels, integrins, the specific integrins are not expressed. And that's why the peptide first accumulates only in the tumor blood vessels, where then they become activated so that they can start spreading. So unless the peptide reaches the tumor, it does not spread. And that's why the peptide is very specific to the tumor.
Okay.
And Annette, if I may add at this point that I think the early work, again, that the hard work done by the scientist team showed that the levels of chemotherapy accumulating in normal tissue was not increased at all by the use of the compound Lister-1. Mm-hmm. And as expected, you would say therefore that the side effects were not enhanced, even though the therapeutic effect was. So to share the experience with our original study, at the 2-year mark, we still had 5 patients who were on chemotherapy, first-line chemotherapy with gemcitabine nab-paclitaxel, who were still responding, no evidence of progression, more than 2 years after starting those drugs. And you just don't see that in normal chemo. And obviously, if this compound was enhancing toxicity, there's no way people would actually be able to stay, on drug for that long. So one of the questions which I've been asked quite frequently is, do I think that because it's enhancing drug delivery, that we may be able to use lower doses of drugs, and in using lower doses, will that actually be side effect sparing? So, will we see less neuropathy with nab-paclitaxel because we can use a lower dose that will still be effective? And I think these are, that there's some fantastic, studies that are yet to be done, in this field, where we may see that it becomes a side effect sparing drug.
So that is another fascinating place this could go. I mean, can you talk to me in the long range? I mean, if this is successful, how do you see it fitting into your armamentarium of anti-cancer drugs? And do you look at it, and is there potential of the drug beyond solid tumors? And I don't know, maybe you as an oncologist are not the ideal person to answer this, but maybe we can take this from both ends. I mean, Dr. Sugahara, during your experiments, do you ever look at this and think, oh, this would be very useful for something else? Or, Dr. Mazzo, what do you have plans or thoughts or hopes for other things that this could be useful for? In general, sort of, what is your big picture plan for LSTA1?
So why don't we start with Kazuki and Andrew, and then I could add a few thoughts after the fact.
Sure.
Okay. Do you hear me?
Yes.
Go
ahead.
You're up.
Hello. Do you hear me?
Yes.
Yes.
Okay. Okay. Yes. In terms of the therapy that can be combined with the peptide, I think there were two questions basically. One is whether we can expand the use for other therapies, and the other is the type of tumor that we're targeting. So in terms of the therapy, at least in preclinical studies, we know that the peptide can be combined with all sorts of therapeutic agents. For example, small molecules, antibody drugs like trastuzumab, also nanoparticles, at least up to like 200 nanometers. Some people have tried to deliver cells, so the gap that's made with this peptide is probably big enough to pass through a lot of different types of therapies. One thing that we haven't really covered is whether the peptide gets into the tumor cell, gets into the tumor tissue, and even inside the tumor cells. And in our translational research experiments, we have evidence that the peptides can also deliver things inside the cells. So that means that we can likely expand the therapeutic agents to also things that need to get into cells. For example, the recent development of KRAS inhibitors or targeting agents that need to get into cells, or RNA-based therapies that really need to reach the nucleus. They can be options that can be combined with the peptide. In terms of the tumor types, we have tested millions of tumor types in mice, at least. And in basically all kinds of tumors, we have seen the effect of IRGD to a different level. In terms of sarcomas, for example, there are good reasons to believe that the peptide would work for them. Those are also rare cancers or rare tumors that need really good therapies. The sarcomas, they are often composed of cells that we know that express high levels of the IRGD receptors, and they also have the blood vessels that the peptide can target. So it makes good sense to target them. Does it make sense?
Yeah. If I may, just add, it's a very interesting problem that's faced to a drug developer when you have a compound that seems to have such a wide potential applicability across multiple combinations and multiple tumor types, and perhaps even multiple disease states. So there's been some work done in inflammation, for example. But I think, in the end, as a company developing therapies that we hope to eventually make available to patients widely on the market, we need to focus. And so, our initial plans have been very keenly focused on proceeding very rapidly in the development of combinations of current standards of care with LSTA1 in pancreatic cancer, because that's where the most work has been done. And there, Andrew and his colleagues have a phase 2b trial, the ASCEND trial, which is running, which is actually quite far along and likely to complete enrollment in the next quarters and have data perhaps as early as next year. Additionally, we are looking at a number of proof of concept trials across a wide variety of other solid tumors initially in order to demonstrate how LSTA1's breadth of applicability is actually translated into the human environment. So we're looking at things like glioblastoma or brain cancer, head and neck cancer, cholangiocarcinoma, and a variety of others in combination with different standards of care, cytotoxics with and without immunotherapy. And we've even begun to explore the possibility of using radioligands in combination with LSTA1 to see if they could be enhanced in terms of targeting and penetration. So the biggest challenge for us as the drug developers with LSTA1 is to remain focused and not to be tempted to go off and try everything on the menu, because obviously that could cause a diffusion or dilution of effort, and also a diminution of capital resources.
So yeah, it does sound like the trialing system you've set up is quite broad. I mean, would you say that is the case? I mean,
Yeah. I mean, and clearly, Annette, what we've done is we've tried to exploit these two pillars of our development strategy. Focused pathway toward earliest possible registration in pancreatic adrenal ductal carcinoma, and then also a variety of solid tumor types, half a dozen or more in different combinations to demonstrate the breadth of applicability. And we've done that in a very capital efficient way. So, it's great to be part of this renowned panel and also to be working with Dr. Dean. Clinically, the added benefit, besides having, a world-renowned oncologist handling the development of the drug, is that he happens to reside and work in Australia. And there are very significant benefits from an R&D rebate perspective for doing clinical research there. And that helps maintain the capital controls on such a program. So that allows a small company like Lisata to affect so many programs. In addition, Dr. Dean is a founder of a charity in Australia, a foundation that is dedicated to finding new treatments for other gastrointestinal tumors. And as part of that, we're receiving some support financially in some of the work that's being done there. So overall, between geographic selectivity, appropriate clinical design, and just very careful management of the clinical programs, we've got a very capital efficient program that allows us a pretty broad, in fact, maybe very broad approach to exploiting the possibilities of LSTA1, at least in solid tumors.
Okay. That is just terrific. Can you tell us a little bit about your background? I mean, how did you come to LSTA1?
Well, I'm a little embarrassed to say, but I'm very rapidly approaching my 40th anniversary in drug development. I've been doing this for quite a long time, and spent quite a bit of my career in executive positions in research and development in big pharma. But I've been in small emerging companies for the last 15 or so years. And I really find this environment very exciting because, big pharma has lots of resources, people, money, et cetera, but there's also typically a lot of bureaucracy. And in small companies, we're much more nimble and we're able to get things done, I believe, in a much more rapid fashion. And when Lisata was formed about a year ago from the merger or acquisition by Caladrus Biosciences of Sen Therapeutics, it gave us the opportunity to really focus on moving this program through clinical development and getting it to patients. And so, it was after several years, literally, of our team scouring the opportunities for new compounds with potential and where we believed our experienced team could bring to bear their expertise to make a material contribution to accelerating the development. And the LSTA1 compound rose very quickly to the top of that list. But as I said, it took more than 2 years to find it.
Okay. All right. So as we've been talking, I've been thinking, and at BioWorld, I covered not just cancer, but most areas of science that are relevant to biotech or could be. So I've got like a blue sky question for you, and then perhaps a more realistic one. Not in terms of ultimate success, but in terms of the developmental path. So blue sky, I mean, one of the things that I have written about and that is sort of becoming more urgent as actual effective antibodies become available for neurodegenerative disorders is that it's just hard to get things into the brain. And it is for different reasons than it's hard to get into tumors, but it is the same principle. I mean, could you see this drug being used, or I would guess not this drug because you wouldn't have the correct binding partners for it, but could you see using this approach to try to get things into the brain? And then not quite as blue sky, but also on the broader front, you're talking a lot about multiple tumors. Would you hope, and do you see it as realistic to get sooner or later a tumor agnostic approval for this drug?
So let me start with the first question, and then I'll defer to Kazuki and Andrew for their opinions on the same question. But we know from preclinical research and some early clinical work that LSTA1 IRGD does pass the blood-brain barrier. Right? But you're absolutely right in that once it's into the brain, the homing receptor it's seeking, the integrins, typically are looking for tumors, solid tumors, and that's why we started a trial in glioblastoma, because we think there could be an opportunity there in combination with temozolomide, for example, to enhance survivability there. But without those integrins present, then the IRGD doesn't really have a role, so to speak, at least this one. So for solid tumors in the brain, yes. For other diseases of the brain, personally, I'm not so sure, but I'd love to hear Andrew and Kazuki's opinion on that. And then we can talk about tumor agnostic approvals, because that's a regulatory speculation that is always philosophical in its nature.
Well, I think, to be honest, David and Annette, being a medical oncologist, I didn't even know there was a world outside solid tumors. So, I mean, I think you're talking Swahili to me at the moment. So, I mean, the thing for me is I just, it's almost like, child in a toy shop where you just got, the world's most amazing new toy, and you have probably having a bit of ADD. I'm thinking about all these amazing applications for across the board for all kinds of solid tumors, but the the thing that's really caught my attention is, so we can say, okay, we can enhance drug delivery to breast cancer, and in doing so, you have a disease that's got a 86, 88% cure rate. We can do it for pancreatic cancer that has, globally about 7% cure rate. And, the reality is we're going to make much bigger impact to the world, for patients, their families, and cancer outcomes by dramatically changing in a kind of disruptive technology way, not a little increment way where you make a small change here and a small change there. I think this drug, I think Lister-1 offers a disruptive approach where we're going to just shatter the previous dogma and paradigms. And if we can really, pump these, get very high levels of cytotoxic drugs into tumors, spare normal cells, and if it really does what we think it does with the immune therapies, then we are creating, perfect storms to get rid of cancers. I mean, the way I explain it to patients when they're coming on study is, we always talk about cancers being parasitic. I would say that, it's no good trying to starve yourself because the cancer will always feed, and the tumor cells are able to take what's in the bloodstream, whether your normal cells get it or not. And, the mechanism is this IRGD, mechanism of the integrins and the neuropilins. And so we are hijacking this point of advantage that tumor cells have over normal cells, and we're using it to feed the cancers, chemotherapy and immune therapy. And so, for my mind, the potential to apply this is, well, pick a tumor and tell me what you can't apply it to. I think it's got so much potential. So I'm excited. Yes, very.
All right. So we've lost Dr. Sugahara. I would have loved to hear his thoughts. Hopefully, there he is. Very good. So we were just saying that we would have loved to hear your thoughts about blue sky approaches for this drug.
Yeah, I apologize. The internet here in my institution is working a little strange. At the right moment, of course. Yes, so various ways of using this drug you're talking about, I think.
Mm-hmm. Yes.
Yes. I heard a little bit about the brain tumor. That is absolutely a very important target, which we believe it's going to work based on our scientific data. And just like Andrew was making a point, it's another kind of tumor that has a very, very low curative rate. A lot of people die from it once you get this tumor. So there's a chance we can make a very big change there. Some other ways of using this drug, in my opinion, is that it can now be combined with certain drugs, not just for drug delivery purposes, but based on our recent knowledge that the peptide can change the microenvironment in certain ways. For example, one example is the immunotherapy, where we can modify the immune microenvironment with the peptide, not only by delivering the drug more, but by changing the character of the stroma. Our recent work has indicated that we can target certain tumors that have more of the receptor for the peptide. For one reason, because we can deliver more drug, but the other reason is that those tumors tend to have a more aggressive character. For example, pancreas cancer that has more of the IRGD receptors behave much more aggressive in the body based on research. So we can basically target those that are worse than better. So I think there are different ways of making use of this peptide, just beyond drug delivery.
Okay. So yes, Dr. Matz, I'd love to come back to you and also ask, again, ask about, as far as you're willing to share, whether, you are hoping, or planning on a tumor agnostic developmental path, but more generally, how is the trialing system you've set up different from others, and why did you do it the way that you are doing it?
Well, I think, to be clear, we're taking a fairly traditional approach initially for pancreatic cancer, at least. And by traditional, I mean that we will be looking at all the guidelines that are available internationally from the FDA, the EMA, and the TGA, the Australian authorities, to determine what is the conservative base case path to a registration, and making sure that everything that we do will meet the requirements to advance through that process as rapidly as possible. But at the same time, we're also exploring all the ways that might be available to allow this compound to come to the patients sooner. And we've all heard about provisional approval pathways that exist in various forms around the world, and we're clearly exploring that. I think it's quite clear that in Australia, for example, the results, if they're positive, as we hope, from the ASCEND phase 2b trial, in combination with the results of the phase 1 trial, might be sufficient for provisional approval application. And we're having those discussions with TGA to see if we can move that forward. That would advance the availability in that territory to patients by several years. And we will plan to have those similar conversations with FDA and EMA, of course, once we have the data. And that, as I said, will be most likely at around this time next year. So those are very important ways to look at it. And, clearly, indication or tumor by tumor is the traditional approach that one would take. And I think, by demonstrating proof of concept, which includes not only the propensity for the product to improve efficacy of standards of care, but also to not exacerbate side effects. I think as we accumulate more data across a wider range of modalities of drugs in a wider population setting of patients, that'll give us at least the opportunity to open those conversations with the regulatory bodies. But I think that that's a second step in the process of getting to market.
Okay. So we're going to open it up to audience questions in just a minute. For my last question, and then I will hand it back over to Sarah and Steve for audience questions. I want to bring it back to the patients, to the individual patients who stand to benefit from these drugs. So Dr. Dean, I'm curious to hear whether you have any individual patient experiences that you want to highlight. And for all of you, what are you hoping this will do for the individual patients? And the very obvious answer is cure their cancer. But I'm thinking, how will this treatment improve sort of the treatment experience? How does it have that potential? And also, as you are doing, as most clinical trials do, you're looking at very late stage patients, of course. What do you think would be the benefits of this drug in patients who are maybe not quite as far advanced as those that are typically in clinical trials?
Well, I think the first thing is to say that the prospect of dramatically enhancing drug delivery to the huge variety of tumors that we believe will benefit from this compound, the potential to keep people on effective treatments for longer, you know, our experience was, as I said, you know, we had 5 people still on trial at 2 years because it was so tolerable. And yes, they'd had, you know, a dose reduction or 2, but at the same time, in having those dose reductions, it maintained its efficacy. And I think that makes perfect sense when you look at the basic science of the compound. So, you know, the potential for me is treating more patients, getting more drug into everybody for longer periods of time, but doing it in a much more tolerable way. So, you know, I mean, that for me is the kind of the holy grail of you know, of cancer treatment. Make it more tolerable, let people stay on effective treatment for longer, and make the treatment work better.
Annette, are you finished asking?
Yes.
Sorry,
no, I wasn't sure whether Dr. Mazzo wants to weigh in on, you know, the individual patients, and if not, yes, I am. That was
okay. Yeah, and I see Kazuki. Yeah, Kazuki was trying to come back in. I think his connection is bad, and I know Erkki tried to come back in. Maybe his powers come back, but in any case, Dr. Mazzo, David, do you want to respond? Do you have a response for Annette?
Only that I would echo what Andrew said. I mean, you know, ultimately we're looking to get something to patients that will make a difference, and that's why we've chosen the cancers we've chosen. As Andrew said, you know, incremental improvement is good, but that's not what we're after. We're looking for you know, paradigm shifts in what patients can expect in terms of, you know, quality of life and survivability, and we think that LSTA1 will offer that, and we want to prove that in the clinic as quickly as possible.
Dave, I'd like to follow it up with one question. What do you expect as a company for the upcoming milestones in the near future, the remainder of the year, and what can we look to see? What's your guidance for us right now?
Well, sure, Steve. I mean, you know, as I've mentioned several times now, we have almost a dozen different programs that are either underway or will be starting very shortly, and a good portion of them will have completed enrollment in, let's say, the next 6 months from now, in general terms. So that means that initial data from several of these programs will be available throughout the year. So we'll see some early data coming out of the Sendifox trial, which is using LSTA1 in combination with FOLFIRINOX, which is the other standard of care that 50% or so of pancreatic cancer patients get if they can tolerate it. And that's also in a few other cancers. Appendiceal is one of them, for example. We'll see some data coming out of China with our partner Qilu, who has been doing some work in phase 1 and phase 2. They've already reported positive preliminary results from their phase 1b, which was essentially a replication of what Andrew and his colleagues had done in Australia, and they've corroborated that and are now preparing to move into phase 2. But most importantly, I think, you know, toward this time next year, we'll see the results from the cohort A of Ascend, which is a randomized, blinded, large phase 2b trial, and that I think will provide, in my mind as a developer anyway, an unambiguous answer to the fact, to the question, do we have a drug here? And what is then the next step to rapid approval? And so that's probably the key point. And then we have a number of other smaller trials, including our glioblastoma trial, a trial in intraperitoneal lavage, et cetera, which all will have data points that will come out in late 24 and in early 25. So the next 12 months or so are going to be very rich with information about LSTA1 in human.
Great.
Okay, so if I can go to some of the questions, and they may be repetitive with some of the discussion. And I think, Dave, you just mentioned that you'll have information on your glioblastoma in 2024. One of the questions was, okay, you're doing it in pancreatic cancer. Do you have any data to share on glio?
Yeah, and as I said, the GLIOS trial is just starting enrollment. It's being done in an area of very high prevalence of glioblastoma that none of us quite understand. It's in
Where was that?
That's in the area, of Europe, near the Nordic countries. So we're actually doing this trial in Estonia and Latvia, but Finland and those areas have higher prevalence than a lot of other countries in the world for some unknown reason. We expect that trial to be in a position to have early data late next year or early 25. And of course, if there are any anecdotal information that we can share before that, we will. But we've taken a very rigorous approach to most of our trials. Most of our trials are blinded and controlled because we want to be sure that at the end, the data can be viewed scientifically as being definitive, as opposed to having been biased or influenced by the exuberance of the people conducting the trial.
Sure. Yeah. Steve, did you say something? No?
No. I'm kind of curious. One of the things that was alluded to, and I see that we've got, I think we have Kazuki back on. I hope we do. Was along the lines of
his name is there. His name is there on the screen.
Okay. Well, my heart is crestfallen. But maybe both of you gentlemen. Oh, here we go. The allusion was to tumors. But this is a terribly strong platform. What other areas systemically do you think might be leverageable if you were to think about it, just whiteboarding? And I'll, I want to end with that because that's a broad question. But just out of curiosity, what are you looking at as far as other areas, if they might come into play? So
right now, we are specifically, not looking at any other clinical programs, and that's a combination of staying focused and getting through what we have with our limited resources, and also being sure that before we put too much more capital into LSTA1, that we have enough verification from human trials that further exploration makes sense. So it's more of a timing question than one of interest. But I do think that Kazuki and maybe Andrew might have some ideas about where the next place to go might be in cancer or otherwise.
So Kazuki, since you keep going off screen, so we want your answer first.
Okay, sorry about that. I'm trying to connect from my phone now. I hope it's better. So, yes, obviously, one thing is to use the peptide for immunotherapy, which has already begun with Andrew. But other areas, what I think is going to be important is to see the efficacy of newer drugs. For example, the drugs that target the signaling pathway, like the recent KRAS inhibitors, are getting really important in different types of cancers. EGF, growth factor inhibitors, things like that, are new types of drugs that we haven't really studied well enough, but have good chance to be enhanced with our technology.
Okay. Andrew?
I think that, again, obviously we're focusing on chemotherapy and immunotherapy, and that's our main program at the moment. But things like enhancing the delivery of the small molecule tyrosine kinase inhibitors, and I'll pick on sunitinib or sorafenib, and looking at how we might increase delivery of these drugs to things like hepatocellular cancer, or which globally is actually quite a big problem. Not so much in the Western world, but it's something that impacts particularly across Eastern Asia, and things like renal cancer, where we don't have any effective chemotherapy, and treatment is essentially it's immunotherapy or small molecules. So I think the ability to to deliver enhanced levels of small molecules into solid tumors is hugely exciting. So if you can put your target molecule into the bloodstream and it's smaller than 200 nanometers in size, then in theory you're going to increase the delivery of that to whichever tumor you're co-administering the Lister-1 with.
I mean, this really sounds phenomenal in terms of its possibility. You hate to get too excited before the proof is in the pudding, so to say. But knowing people that have succumbed to solid tumors, this is really, really important work. One or two other questions before we sign off. I know we're getting very close to the end. Have you seen any evidence that the tumors become resistant to the therapy? This is a question that came in from the audience.
Yeah, that's really interesting. Well, the work that we've done so far has been very much, first-line therapy. So where we have used it has generally been incredibly effective. So one of the questions that I was super keen to answer, which is whether, if a patient, if the patient's cancer becomes resistant to first-line therapy, could we change to second-line therapy, continue Lister-1, and expect to get just as much enhancement second time round? And my belief from the science is absolutely. There's no reason to think why it shouldn't work. But I would certainly ask Kazuki what his view on that would be. Kazuki.
Yes, I'm back.
I hope you
I agree with Andrew that for now we have no reason to believe that there will be a strong resistance building up to the peptide. I think, as far as we know, the peptide works based on the expression levels of the receptor. And usually these receptors, once used, they are recycled, meaning that even if it goes down, it comes back up after some time. So as far as that keeps going, then we should expect that the therapy keeps going too. And I think that's partly shown by Andrew's earlier work in the phase 1 trial, that a lot of people kept responding to the peptide.
Okay, thank you. And one last question, and this may be a little bit outside of the scope of the discussions. One of the questions that was asked is, will the production of this therapy be really expensive?
So I can speak to that. The answer is...
Yeah, you're the CEO. You know.
Yeah, but I'm also I've also been doing manufacturing of drugs and drug products for a long time. Okay. And, at least in that perspective, the answer to that question is no. But of course, different people who are not necessarily familiar with drug production costs may define, really expensive differently. So I would say that this compound will clearly be somewhat more expensive than most traditional small molecules, but nowhere near as expensive as most biologicals, and orders of magnitude less expensive than most cellular therapies. So, cyclic proteins of 9 amino acids, that kind of solid state or even liquid phase synthesis and manufacturing of the drug substance is chemistry that has been around for decades. It's quite straightforward. It's very well controlled. And, and so the cost of goods here will be, very reasonable from drug perspective terms. And then the drug formulation, the drug product itself is very simple. It's just the lyophilized product reconstituted in a simple buffered aqueous solution. So nothing fancy there, and then sterile fill into vials. So, this is all kind of, if I can use the term, plain vanilla from a parenteral perspective for drug products.
When it comes to drug development and drug manufacturing, plain vanilla is pretty good, right?
Right.
So that means that you would have no problem manufacturing the quantities that might be needed for large populations.
No, we've actually in the process of we've already scaled up, and we can make, you know, kilos, tens of kilos, in fact, of the drug substance, which could translate into you know, tens of thousands, approaching hundreds of thousands of doses of the drug product, and you know, and the scale up is gone quite well as expected. So yeah, we wouldn't have any trouble manufacturing and meeting the needs. That would be a really good problem to have, in fact.
That would be. Be.
So that's enough for me for next month, Dave.
That's right, Andrew. Keep sending your requests.
So wonderful. I think that's it. I think we're out of time. Yes, we're a little bit over. So I do want to thank you all for thank the viewers, number one, and thank you, Steve, for being the sponsor of this event, Annette for leading the discussion, Kazuki for trying and trying and trying again to come back on screen. And please tell Erkki we noticed that he tried to come back on screen, and we really appreciate it. Andrew, thank you for joining us. I hope you can go to bed now and think about all the ways you're going to use the new quantities of therapeutics that you'll get from David. And David,
I shall dream about nothing else.
I am sure you will. So I also, before we sign off, want to thank the audience for joining us today and for submitting your questions. We'll see you again at our next virtual salon. And if you can be in New York for BioFuture, join us at BioFuture October 4 to 6. I promise the UN will be gone by then. The Clinton Global Initiative will be gone by then. You can get through the streets of New York. Thank you all.
Thank you.
Thank you.
Thank you.
Thank you.
Bye-bye now.